Hello and welcome to this webinar, Kill the Tumour, Spare the Healthy Tissue. Ultra-high content imaging enables target discovery for CAR T-cell therapy. I'm Lou Rahman, Editor-in-Chief at DDW. Today's webinar will be presented by Dr. Christoph Herbel, Miltenyi Biotec. CAR T-cell technology provides the foundation for established immunotherapies against certain types of blood cancer and holds great promise for the fight against many solid tumours too. One drawback of conventional CAR T-cell technology, which relies on a single target antigen, is the lack of tumour-associated antigens, TAAs, that are truly specific for cancer cells. This means that the cytotoxic CAR T-cells not only kill the tumour but also damage healthy tissue. One way to improve specificity is using a combination of TAAs expressed on the tumour but not on the healthy cells. Dr. Herbel will present a newly developed workflow to identify new tumour markers using unbiased, high-throughput flow cytometry-based screening and multiparameter imaging. To assess the safety profile of potential CAR targets, Dr. Herbel's team analysed the marker expression across many healthy human tissues using high-content imaging on the MACSima Imaging Platform. Just some background before we start on Dr. Herbel. Dr. Herbel is a team coordinator at Miltenyi Biotec, where he identifies and validates new targets for immune cell-based cancer therapy using the MACSima Imaging Platform. He performed his doctoral thesis at the German Cancer Research Center, investigating the crosstalk between DNA damage response, metabolism, and epigenetics. Fascinated by this interplay of key cellular processes, he started a postdoc at the Dana-Farber Cancer Center studying metabolic signaling pathways that influence cellular responses to genotoxic stress, and he joined Miltenyi Biotec in 2017. Okay, Christoph, over to you now for the presentation.
Thank you for the kind introduction. Hello and welcome everyone. My name is Christoph Herbel. I would like to start thanking the organizers for this opportunity to present our target discovery and validation workflow today. Let me guide you through today's agenda. I would like to begin with a brief introduction into solid tumors and chimeric antigen receptor T-cells, or short CAR T-cells. We will continue with an overview of our target discovery workflow, followed by a description of our ultra-high content MACSima Imaging Platform. Thereafter, I will guide you through some examples of solid tumors, namely glioblastoma, pancreatic and ovarian cancer, which we applied in this target discovery workflow. I will show you subsequently preclinical data for CAR T-cells addressing the newly identified targets. After that, I will conclude and give you an outlook. Let's get started with a brief introduction to solid tumors, challenges and opportunities related to CAR T-cells. CAR T-cell therapy has been very successful in liquid cancers. However, solid tumors are the new frontier in CAR T therapy. New challenges such as the tumor microenvironment and accessibility to the tumor in combination with previous known issues like tumor heterogeneity will ultimately affect CAR T-cell therapy efficacy, and it will potentially raise further or novel safety concerns. The generation of CAR T-cells is a multi-step process. It starts with the isolation of T-cells from donor-derived blood. Next, T-cells are transduced and expanded ex vivo, either in a lab or in a dedicated manufacturing facility. The aforementioned process steps can be automated to reduce manual handling. After ex vivo cultivation, the final CAR T-cell product is reinfused to the patient where CAR T-cells will detect and destroy cancer cells in a target-dependent manner. CARs are similar to regular T-cells and combine the TCR signaling in a single molecule. A major advantage of CAR T over TCR is its MHC-independent antigen recognition. Thereby, tumor immune escape mechanisms can be overcome. By addition of further genetic modification, additional features can be assigned to the T-cell, for instance, to overcome the immunosuppressive tumor microenvironment. In order to design an efficient CAR construct, one has to take several considerations into account. You may consider this triage of indication, target, as well as technology as framework for CAR design. The indication of choice has a strong influence on the target and also determines other means that might be addressed by dedicated CAR technologies, for instance methods to overcome the tumor microenvironment. Coming to the overview of our new target discovery and validation workflow. which is a three-step process: flow cytometry screen, multiparametric cyclic immunofluorescence analyses, and accompanying bioinformatic analyses. We started with our antibody library to identify target candidates, and we use patient-derived xenografts or primary human tumor samples as starting material. Here's the same workflow at a more detailed view. In the first step, we dissociate the tumors to single-cell suspension and performed a flow cytometry-based cell surface screen. These resulted in a first selection of target candidates supported by bioinformatic analyses. In the subsequent steps, the target candidates were validated on the respective tumor samples. In addition, we analyzed the target candidate expression on healthy human tissues to address on-target of tumor reactivity, which can be considered as a potential safety issue. Data analysis is based on cell segmentation and subsequent data extraction, correlation, and clustering. After analyzing expression levels, target heterogeneity, and further spatial information from tumor and healthy tissue, we nominated top candidates for further functional characterizations. Let me choose the MACSima Imaging Platform, which is central for our workflow. Here we have the MACSima device itself, complemented by consumables and reagents, as well as the image analysis software. The MACSima device is a fully automated system including the liquid handling, sample and reagent staging, as well as the microscope. In contrast to classical microscopy, where you are limited to a 5-plex, the MACSima system can achieve over 200 plex. This high level of plex capacity depends on a cyclic approach, consisting of staining, imaging, erasure, and repetition of this cycle. Looking at this in more detail, you would start with a blank image, do your staining, take an image, you erase, blank image, you stain, you image, erase, and you continue, and you recycle and recycle over and over again. And in contrast to classical 3-plex images like you have it here, you end up now with a stack of images of the very same area of interest. And as depicted here by this little movie, you can see that each single image of that slide resembles one marker of interest that is stained. Here you can see a series of images derived from the little movie we've seen before, which come from the very same region of interest that have been imaged. And as depicted here, you can see several tens of markers which we can increase in complexity up to several hundreds. As pointed out, I will talk about target discovery and validation. However, you can imagine several other applications where high-dimensional analysis is key. For example, deep phenotyping of cells or biomarker research and discovery. Coming now to the work we have done to identify new targets and target combinations. Here I'm showing you the workflow again. Flow cytometry-based screening of PDX models or primary tumor, employing our antibody library. Validation of selected candidates by mix on tumor tissue and healthy tissue, which was then further analyzed as shown. I would like to start with the first solid tumor model, pancreatic cancer. In this case, we have analyzed 17 different PDX models of pancreatic cancer using our antibody library with more than 350 candidates. Thereby, we reduced the number of candidates to 50, which we ranked by informatic analysis of publicly available datasets. These candidates were subsequently validated on 2 primary pancreatic cancer samples and additional 7 pancreatic cancer samples via flow cytometry. Thereby, we identified our 4 targets of interest: CD18, CD66C, CLA, and TSPAN8. Here's an example of 3 markers identified by flow cytometry as potential target candidates: CD59, CD318, as well as CLA. However, imaging-based validation of our markers of interest revealed different findings. On the one hand, one potential candidate, CD59, is strongly expressed on non-tumor cells, highlighted here in red. On the other hand, CD318 as well as CLA nicely co-localize with epithelial tumor cells, depicted here and right here. After segmentation and data extraction from the image stacks, we were also able to perform cluster analysis and identify different cell types within our tumor samples, such as tumor cells, B cells, T cells, and many other cell types. Interestingly, we identified several tumor cell subclusters as depicted over here. After target selection, we generated various CAR constructs specific for the different targets. These were characterized in vitro, and the best CAR lead candidates were subsequently applied to immunocompromised NSG mice bearing cell line-derived xenografts. Here we analyzed xenografts from mice treated with two different CAR constructs. In particular, we investigated CAR T-cell infiltration in the tumor, tumor remnants, target expression, as well as macrophage infiltration. We found one candidate, anti-CD66C CAR, that was not able to infiltrate tumor, here depicted by the lack of LNGFR positive CAR T cells, and thereby tumor remnants were still present in the xenografts expressing the target of interest. Moreover, these CAR candidates were not able to recruit murine macrophages to the tumor site. On the other hand, our second CAR candidate, anti-CD318 CAR, was nicely localized within the tumor, depicted here by the double positive CD4-CD8 LNGFR cells, which almost completely eradicated tumor cells with barely 318 expression left. Moreover, these CAR constructs were able to recruit murine macrophages to the site of action within the tumor. Let's switch gears now and change the solid tumor indication from pancreatic cancer to glioblastoma and ovarian cancer, respectively. We performed the same target discovery workflow as described for pancreatic cancer for the two other indications. Here, we are focusing on the on-tumor expression of target candidates. For glioblastoma, we confirmed expression of known targets, such as GD2, EGFR, and c-Met in several patient samples. On the left, you can see the different expression patterns of the three different markers in two patients, GD2, EGFR, and c-Met. In the case of ovarian cancer, we identified a subset of patients who co-express a novel combination of EpCAM and Thy1, also known as CD326 and CD90. These two markers are often used to discriminate epithelial cells which express EpCAM from other cell types such as fibroblasts, immune cells, or hematopoietic cells, which are supposed to express Thy1. On the right side, you can see three different patient samples with different co-expression patterns of EpCAM and Thy1, reaching from a complete co-localization to something down almost 50%. In order to analyze off-tumor expression of our targets and target combinations, we performed mix analysis on a variety of healthy human tissues. In this first panel of tissues, you can see that the target combinations are very specific, with few or no cells co-expressing the marker combinations. In the second panel of tissues, you can also see that the target combinations are very specific, with few or no cells co-expressing the marker combination, highlighting safe target combinations. Moreover, we quantified the expression of our targets on segmented cells over all healthy tissues analyzed. You will appreciate that the relative expression of the markers vary between the markers and between the different tissues. Quantification of the target expression on segmented tumor cells confirms elevated expression of some known markers like GD2, EGFR, and c-Met for glioblastoma, but also for the new pancreatic cancer targets CD66C, T-span8, or CD318. In addition, in ovarian cancer, we also see elevated expression of EpCAM as well as Thy1. This analysis, in combination with correlation and cluster analysis, guided the decision for combinatorial target pair selection. These are the targets that we will follow up in the coming functional assays. For pancreatic cancer, we decided on a target pair consisting of CD32018 in combination with T-SPAN or CD66C. For glioblastoma, our selection was GD2 and EGFR, and for ovarian cancer, we decided for the novel target pair EpCAM and Thy1. Coming to functional CAR assessments, classical CAR T cells have a direct recognition of its respective antigen, and it's in a constant on mode. We apply a different CAR concept, the adapter CAR. This system is a modular system and very flexible. The adapter CAR itself recognizes a tag moiety. The target specificity is achieved by an adapter molecule, which could be an antibody, a Fab fragment, or an antibody, for instance, which carries a tag that is recognized by the adapter CAR. This could be biotin, a FITS molecule, or any other chemical or recombinant tag. The system allows flexible approaches with dosing via titration of the adapter molecule and very easy multi-targeting via the addition of different adapter molecules at the same time. Thereby we can also generate a switch between an off and an on mode of the CAR T cell. A common issue in immuno-oncology is the identification of unique cancer targets that are exclusively expressed on tumor cells. Another common approach is the use of tumour-associated antigens. These antigens share some expression with non-tumor cells. Therefore, it is critical to increase the safety of these targets. And one way is simultaneous multi-targeting of several antigens. Multi-targeting with the adapter CAR system is relatively easy and flexible by applying multiple adapter molecules at the same time in combination with the adapter CARs. In addition, the adapter CAR system offers the possibility to use low doses of multiple adapter molecules. Thereby, each adapter on its own is insufficient to trigger T-cell activation. However, the sum of multiple low-dosed adapters can overcome the activation threshold, leading to cytolytic activity of the CAR T cell. Thereby, we increase the therapeutic window as well as the safety profile of our targets. Moreover, the adapter CAR system simplifies the CAR generation and manufacturing, since the basic CAR construct can be used in a universal way. Initially, we analyzed the novel ovarian cancer target combination EpCAM Thy1 with the adapter CAR system in vitro killing assays. You may appreciate that the two adapter molecules targeting EpCAM or Thy1 respectively can be modulated by the titration of the adapters. Here you can see titrations from 1000 to 0.1 nanogram per ml concentration of adapter molecule, and you can see here that only at high doses the adapter system works. However, using the EpCAM adapter molecule, you see a much wider range of activity. The anti-EpCAM and Thy1 adapters simultaneously, a titration effect can be observed too, as you can see here, titrating different amounts of between 1 nanogram and 1 picogram per milliliter of the adapter molecules. Interestingly, when we compare the single adapter doses with the respective combination, here dedicated with 0.1 nanogram per ml, in the Thy1 or in the EpCAM combination, you can see a strong increase in comparison to the single adapter molecules. Thereby, we could demonstrate that we can improve the therapeutic window. With this, I would like to conclude on our target discovery workflow using mixed technology. We analyzed single-cell expression of hundreds of proteins over a variety of tissues. We were able to identify novel combinatorial targets for different solid tumors. Finally, we applied the adapter CAR system for multi-targeting of the novel EpCAM and Thy1 target pairs for ovarian cancer. For the future, we can envision that patients will be screened for target expression to personalize cancer therapy. Having a panel of adapter molecules against common targets, this will allow to mix and match the adapters for each patient individually. After therapy, potential recurrent tumors might be screened again and therapy might be adapted. At the end, I would like to thank you for your attention and our collaboration partners for their support. With this, I would like to end. I will be happy to take any questions.
Thank you, Christoph, for that presentation. I'd just like to point out before we go ahead to the question section that there are some handouts which accompany this webinar, and you can download them from the handouts area on the control panel. Before we continue to questions, I'd just like to remind everyone that this webinar will be available to access on demand on the DDW website, www.ddw-online.com. I'll now pause for a few moments to allow audience members to send through any questions that they might have for the presenter. Okay, Christoph, are you ready for questions?
Hello everyone, ready. Good to go.
Okay. The first one that we have is, why did you not perform the 400 antibody screening which you performed in flow with the MACSima platform?
Yeah, no, that's a good one. So basically what you have to do is you have to make a trade-off between in-depth characterization and at the same time how many tissues are you interested to look at. And thereby we just had to make a trade-off and decide to take a reduced number but increase the number of samples that we analyze.
Okay, thank you, Christoph. And we've been asked, is there a limit to the amount of markers stained with MACSima, and if yes, what is it?
Yeah, so far we tested a variety of samples and fixation methods which all play into that setting. But so far with PFA fixation, we reached 400 plex, basically. Yeah.
Okay. And what are the modes of signal erasure of the MACSima?
So basically, we are currently using two technologies. So either we work with photobleaching, that is one way to erase a signal, or with the enzymatic reaction to cleave off the dye.
Okay, thank you. So the next question is, how do you compare the novel targets you introduced to known targets, such as mesothelin, CD133 or MUC1?
And that's a very good one. Yeah, we also found these new known targets, which we could also validate with bioinformatic analyses. And yeah, so in terms of inter-patient heterogeneity as well as intra-tumor heterogeneity, our targets compare pretty well with the known ones. And we also saw some nice correlations with the known targets in the field.
Okay, thanks, Christoph. We've also been asked, what's the maximum field of view that I can analyze?
Yeah, so basically it's rather broad, a couple of millimeter by millimeters that we can image. However, that comes also with a trade-off. So you have to basically take into consideration the larger your field of view is, the longer it takes to image that area. So you have to make a trade-off, but you can pretty much image rather large regions of interest.
Okay. And how many samples can be analysed at once?
That depends on the disposable that you're using. So that can vary between 1 sample or 24. And we are also, with our Maxwell 1 frame, you can also use TAAs, for instance. And you have one class slide and you spot 50, 60 tissues on it. So you can also try to multiplex with such a system.
Great, thank you. And we've also been asked, can I also use my own antibodies, or do I have to use the ones from Miltenyi?
Yeah, okay, yeah, that's a very fair question. So you can use your own antibodies. You only have to consider that it's suiting the protocol of the MACSima device and do some validations. That we provide you with, they are validated for the mixed technology, but you can definitely use your own ones for sure.
Okay, lovely. And how is it when it comes to designing and validating antibodies against new targets of interest?
Yeah, so you have a couple of options. So we have either pre-selected panels that you can pick from. You can use single antibodies, design it on your own, or you can also consider to use entire blades with all the antibodies available. So yeah, so these are the options basically. So you can either have the full coverage, you cherry pick like pathways or modules that you're interested in, or you just pick single ones that you are interested in.
Thanks, Christoph. And then we've also got another question here, which is, although you reduced considerably your panel of antibodies to run on the imaging platform, what was the most challenging aspect of performing a run with 100 markers? They've said it can be so difficult to optimize staining protocols just for a couple of markers.
Okay, let me see what I get it. So the most challenging aspect we had in the past was basically to find this balance between the tissue, the fixation method, and a proper panel of validated antibodies. So bring all these pieces in place and balance them. I think that was the most challenging part in the end, but very, yeah, surprising.
Okay, lovely, thank you. That's all the questions that we've had in there, Christoph, so thank you for that. Thanks for those responses.
Thank you. It was great joining you today.
Thank you. If you'd like to listen to the webinar again, it will be available on demand at www.ddw-online.com. You can now view Drug Discovery World content online on your mobile or tablet or via the DDW app, which can be downloaded via the App Store or Google Play. Thank you for listening.